Hi, and welcome again to a new video here to review our human physiology and the classes we went over through with you at Florida Gulf Coast University. In the next two videos, what I will do is I will try to review together with you the immune system. As we discussed at the lecture, we have two major components in the immune system. One we're born with, and the other one is what we call the adaptive system. We also agreed that the one we're born with is non-specific, and we call that the innate system. We discussed the components. And what we're going to do now is to review what we learned about the innate system. And then in the next video, I will discuss with you the adaptive immune system. We're going to start with the humoral immune system, or the humoral immunity, and that will involve B lymphocytes, which become plasma cells, and these are the guys that secrete to you tons and tons of antibodies. And then we will go into the cell-mediated immunity and the T lymphocytes, the cytotoxic T cells, the Treg, the regulatory T cells. Let's start first with, like I said, with our innate immune system, to review again, or to remind each other of the blood cells that we will be talking about in this chapter. We will be discussing a lot the neutrophils. These are your bacteria slayers in your blood, and it's highly phagocytic cells. But they're not phagocytic when they are in the blood, but once they migrate into the tissue, they become active at the line of duty to interact with the foreign invasion, or if there is tissue damage, and we will see how that happens. We will also talk about the monocytes, which give rise to the macrophages in your tissues. Once they migrate from the blood into the tissues, they will differentiate and they will become what we call macrophages, and different kinds of macrophages. Basophils, we're not going to discuss it in great details in this lecture, but we will talk about it especially when it comes to the hypersensitivity, because they are associated with a lot of the adverse effects we see with hypersensitivity and allergic reactions. Eosinophils were primarily, we think they were made to fight parasites, and now that we don't have parasites, we think they are also associated with some hypersensitivity and allergic reactions. The lymphocytes, like I said earlier, we will discuss both branches, or both types, major types of the lymphocytes, the T lymphocytes and the B lymphocytes, and let's not forget, of course, the natural killer cells being as well lymphocytes, although the natural killer cells are not part of the adaptive immune system, but rather they are of the innate system, as we will see later on. The erythrocytes and the platelets, we're not really going to discuss them in the immune system, but when we are talking and when we are discussing the blood chapter, we will review those together. So again, just to summarize things that we will cover again when we get to the blood chapter, I want you to realize that the lymphocytes are originating from a certain lineage of the hematopoietic stem cell, and that will give rise to lymphoid stem cell. In the meanwhile, the myeloid stem cell, on the other hand, will give rise to a lot of other cells, the cells that will give you the platelets, the cells that will give you the erythrocytes, or what we call erythrocyte precursors, or erythroblasts, and erythroblasts will be somewhere here. And granulocytes, those will give you the basophils, eosinophils, and the neutrophils. Any cell that has granules in them, we will call these guys the granulocytes. And then we have everything else, the agranulocytes. So this is an agranulocyte, the monocyte is agranulocyte, and the lymphocyte is, of course, is agranulocyte. So that's another kind of classification we can base our bloods at. But I just want you to remember from this graph the lineage of the lymphocytes and the lineage of the granulocytes, because often we hear the term myeloid leukemia, acute myeloid leukemia, acute lymphocytic leukemia, and I want you to understand where they come from. They're essentially cancer of either of those stem cells. Okay. So, as we discussed, the lymphatic system is composed of vessels. The lymphatic capillaries will drain the extracellular space, and that will be carried by larger vessels. It will go into a cleaning station, or lots and lots of cleaning stations. We call these guys lymphoid tissues, and these will be your lymph nodes. These are the primary. They're not primary, they are the secondary, actually, lymph tissues. The primary lymph tissues we will be talking about will be really the thymus and the bone marrow, since the synthesis takes place there. Everything else is secondary, and that will can include the lymph nodes, the mucosa associated lymph tissues, or what we call MALT, your tonsils, even the spleen. All of these guys will be referring to as secondary lymphatic tissue. These are very important components, as we will learn later on, especially when we discuss the T lymphocytes and the B lymphocytes, because many of our lymphocytes will be hunkering down in those stations and waiting and anticipating action in order to give you the adaptive response that you're looking for. And we will cover that in our, not this lecture, but the following lecture. And from here we move on to discuss then the immunity. What's immunity? Immunity would be defined as the resistance to a disease. As I said earlier, the immune system has two intrinsic systems. One of them we will call the innate, or what you're born with, defense system, and the other one is the adaptive defense system. We will realize that the adaptive system has certain characteristics. The fact that it is highly specific, and it is systemic, and in most cases it has memory. We'll discuss that, and that is very different than the innate system. And as you remember in the lecture, we compared the innate system with the patrol officers who don't have a picture of a suspect, but they're looking around neighborhoods, dealing with anyone that would sort of fit the general criteria of being suspicious or a criminal. Whereas a highly trained detective or an assassin or something will have a picture ID, and they are only looking for that particular suspect, and they're not bothered by any kind of problem surrounding them. They're only looking for one suspect and one suspect only, and that will be part of the adaptive system. So, moving along, we'll talk about the innate defense system to be your primary line of defense, the first line of defense before it calls upon the highly adaptive immune response that you will have later on. you will have, before you even get to the tissues and dealing with this with your innate system, you will have walls that will defend you. You will have intact skin, you will have mucosa, you will have acids, you will have secretions that are hostile to organisms and pathogens. You will have enzymes like lysozymes being secreted in your sweat, as part of your sweat or as part of your saliva. You will have digestive enzymes in your intestine. You have mucus that will stick to any pathogen, at least most of the pathogens, and dust. And along with the ciliary movements from your respiratory tract lining, you will be brushing out that mucus that is now trapping bacteria, viruses, dust, all kind of particles outside your trachea, and then you are subconsciously swallowing that, and it will end up in your stomach, and it will be subjected, of course, to very harsh treatment there. So this is the very first line of defense which we discussed. The skin, the mucous membrane, we have even hair in the nose that will trap and filter dust and particles, and we discussed the cilia and the fact about the mucus that is secreted by your lining, your mucosal lining, and that of course has a very important function to trap your particles. Again, we will have fluids. These are hostile fluids to any invasion. They're mainly acidic. The lacrimal secretion has lysozymes, and it continuously washes off your eye. The saliva, the same thing. We have also lysozymes, and it cleans the mouth. The urine will flush the urinary system. The vaginal secretions generally acidic, and they make the life of organisms that want to grow in that area very hostile as well. We will have chemicals like oil from the sebaceous glands. We will have the gastric juice, the vaginal secretions. All of these guys are acidic, and that of course again will make things very hostile environment for pathogens to grow. Now, when things don't work the way it is supposed, if you have a break in the skin, or you have a virulent organism that can bypass all these natural barriers and get to the underlying tissues, then we will activate our innate immunity, and then we will have those four components that you're looking at on the screen. Namely, we will have inflammation. We'll talk about each and every one of them. Inflammation, interferon, natural killer cells, and the complement system. Now, as I said in the lecture, before we get into the natural, the innate immunity, let's discuss together the players that are associated with your innate defense system. So let's talk about the macrophages and the phagocytic cells. We have major players here. One of them is the macrophages, are the macrophages, and these macrophages, as we discussed earlier, they have migrated from the blood. They used to be monocytes, and once they are now in your tissues, we'll call them macrophages. They assume different shapes, and but they all have the same function, which is eradication of a pathogen, or eradication of a dead tissue, part of a dead tissue, some cells that died, and there is of course constant regeneration, apoptosis and regeneration that happens throughout your life, at least in some tissues. Now, you have different types of macrophages. Some of them, they wander around, like the ones we saw in the alveolar macrophages. These can essentially walk on the alveolar walls, and they will be like patrol cars going back and forth in the neighborhood looking for action. Now we have others that are fixed, like the dendritic cells and the microglia in the brain, and they're not really moving around. They are sitting there and again waiting for action. But interestingly, once, for example, the dendritic cells are called upon and they have phagocytosed whether it's a pathogen or dead tissue, then they will mobilize and they will leave your tissue. But before they do so, they will send a signal, like an alarm or SOS signal, to bring in more phagocytes, to bring in new monocytes, which will become macrophages, and to even send a message, as you will see later on, to your hypothalamus to increase your temperature. They will send messages to your bone marrow to make more phagocytic cells, mainly neutrophils. So they have lots of functions, and then once they leave the tissue, they will go to your lymphatic tissue, lymphatic circulation. They will go to the lymph tissues, the lymph nodes, where they will start to interact with T lymphocytes and the B lymphocytes, and you will see how they play a very critical role in the activation of your immune system and the adaptive immune response. So that's a little bit, couple of words about the macrophages. And the other phagocytic cells are the neutrophils. You have to distinguish between the two. The macrophages, of course, they have their own lineage and they are coming from the monocytes. Neutrophils they have been neutrophils, and they are coming from the blood as well. And we will see how they migrate from the blood into the tissues, and in a process we call diapedesis. We'll see how this immigration takes place in the next few slides. So the neutrophils, they're not phagocytic when they are in the blood, and we know them, and we will see that in the blood section when we cover that. They are very characteristic by the fact that their nuclei, or the nucleus, looks as if it's divided into many nuclei, but they're not. They're connected with tiny strings, and the lobules, or the small parts of the nuclei that are connected together, it looks like three components or three lobules are connected together, and that's why we call it polymorphonuclear leukocytes. The leukocytes, the white blood cells that assume different shapes of the nuclei, they don't have a particular shape, but like I said, it looks as if the nucleus has been divided into like three components. So we have a video here, and I showed that video to you at in the classroom, and where one lymphocyte in this video is chasing down some bacteria and phagocytosing it. And I do want you to realize that it is an active process. The lymphocyte, not the lymphocytes, the neutrophils are not going to just hang around and wait for the bacteria to show up at their at their doorsteps, but indeed they're going to go and they will chase that pathogen, and until they win the battle and they do engulf that pathogen. And what you also need to know is that when you have a pimple or a little suppurative infection, the pus you see is actually composed of neutrophils. These are white blood cells that died while trying to eradicate your problem. And you will see how they do that, and the kind of enzymes they secrete, and the kind of oxidative burst they will try, and even they make some bleach in order to destroy your organism, the one that they engulfed. So they are very effective indeed, and you will see how they do their action. But I want you just to remember this video and see how they chase down the bacteria. And this is a process we call chemotaxis, where the bacteria is releasing some chemicals, and those chemicals will become very appealing to the neutrophils, and they will chase down the source of those chemicals, and therefore they will be able to destroy the bacteria. Think of them as a tag dog that sniffed a target, and now they are following that scent to hunt down that particular target. All right, so what does phagocytosis serve us in? Well, you are destroying pathogens, and we will see how do you destroy pathogens initially by lysosomal enzymes and also by acids. Where are these guys? They are in vesicles inside the neutrophils and inside the macrophages, and these vesicles are called lysosomes, if you remember from biology classes. Now, your neutrophils are also capable of producing what is known as respiratory burst. And in the respiratory burst, you have a release of H2O2, and also the release of bleach or hypochlorite, and you will see that in the next slide. The neutrophils are capable of making defensins, and the defensins are essentially antibiotics that are naturally occurring, and they will interact with your organisms and destroy them as well. So if we think a little bit more about the oxidative or the respiratory burst, the neutrophils are capable of utilizing an enzyme called NADPH oxidase, and that will make H2O2. The myeloperoxidase will take H2O2 and chloride to make hypochlorite, which is the bleach. And of course, when you don't have these, then you have repeated infections, because your innate immune system is not capable of dealing with the problem as it should. This is not just limited to the neutrophils. Also, the macrophages are capable of producing the oxidative bursts. How do the phagocytic cells, whether they are the neutrophils or the macrophages, how do they identify their target? Remember, these are not specifically targeting a particular organism or bacteria or a virus or even a cancer cell, but instead they're looking for a general criteria, something that doesn't fit the normal. And whether this pattern is associated with pathogen or it's associated with damage, in either case, those phagocytic cells will be able to sense their target by some receptors. We call these receptors toll-like receptors, or TLR for short, and they will recognize the pathogen associated molecular pattern or the damage associated molecular pattern and go after them. So here's the story. You have with phagocytic cells, they will form a phagocytic vesicle or a phagosome that will fuse with the lysosome, and then the acid hydrolase enzymes, they will start to break up those foreign matter, whether it's protein or dead tissue or whatever it is, and then with the process of exocytosis, you will be expelling that. This is true for the neutrophils, but for the macrophages, we're going to add something else, as we will realize later on, and it's extremely important, very, very, very important part of your immune defense system, something we call antigen presentation. Now, the macrophages and the B lymphocytes are capable of antigen presentation. What is the antigen presentation? It's the fact that after a macrophage would destroy their target by phagocytosis and digestion, they will keep some of their peptides and they will present what they destroyed, pieces of what they destroyed on their surface on a certain protein called MHC, class 1 MHC. And we'll talk about that later, but I just want you to appreciate the fact that macrophages, dendritic cells, and also the B lymphocytes, all of them are capable of what we call antigen presentation. And that will become, this kind of information will be very, very, very important later on as we proceed in understanding our immune system. Okay? So, as I said, we have 4 components of the innate immune system. Now, we are starting now with the natural killer cells. We will move from there into discussing the inflammation and the complement system and the interferon. We will discuss all of these together. Now, the natural killer cells are not phagocytic cells, but they are either gonna destroy the cells by contact, where they will put a hole in the cell and they will inject enzymes that will destroy the cell from within and induce apoptosis, or the natural killer cells will also secrete some chemicals that are capable of killing the cells in their surrounding, in their vicinity. Now, the natural killer cells, again, are not specific. They're looking for non-specific targets. They're looking for cells that are lacking MHC, class 1 MHC, or they have abnormal class 1 MHC, and which are events that take place when the cells are hyperactive, like in case of course of cancer, or in case of if the cells have been hijacked with viruses, then the production machinery inside the cells is being hijacked, and therefore the cells don't have enough time to make MHC. In either case, when the cells have unusual MHC, then they will become a target for the natural killer cells. In other words, the natural killer cells now will be capable of destroying cancer cells. And it's no wonder that the natural killer cells are becoming now a very, very appealing target for cancer therapeutics. And we can excite those cells, and you will see, like interferons, for example, are capable of exciting them. The natural killer cells can be excited with chemicals that are coming from the macrophages, but at the same time, the natural killer cells will also excite the macrophages to make them really nasty macrophages, macrophages that are capable of much more destruction. So they talk back and forth to your other cells, and whether those cells are part of your innate response or adaptive response. So I think I have a video that I will show to you later on about the natural killer cells. To define them, they're cytotoxic cells. From their name, it shows they have granules, and these granules are rich in what we call perforin. Those are like daggers or arrows that will perforate the cell surface of the cells, whether the cells are viral infected cells or the cells are cancer cells. They also will secrete protease, and from the name, it digests proteins. They express CD16, you don't have to remember that part, and that will aid in the antibody-dependent cell-mediated cytotoxicity by activating the B lymphocytes. So, but that part you don't necessarily have to remember. The natural killer cells are activated in response to interferons, and they're also activated by cytokines, which are chemicals or paracrine factors that are derived from the macrophages. The natural killer cells are going to secrete interferons and tumor necrosis factor, and as you will see, tumor necrosis factor is capable of much more things. It's capable of activating the macrophages, it's capable of stimulating the lymphocytic proliferation, and as I said earlier, the natural killer cells are very, very heavily involved in what we call tumor cell surveillance, looking around for tumor cells in your tissues or in the blood if the cancer has already found its way to the blood. But unfortunately, the cancer cells sort of figured a way to evade that problem. The same thing with the viruses. What these cells are capable of doing, cancer cells and viral infected cells, they make a protein that looks like MHC, and therefore the natural killer cells will think they are normal cells, and they will just walk by them without destroying them, without destroying the targets. So again, the natural killer cells are very heavily studied nowadays to provide us with a new hope for treating cancer. And several studies, very successful studies, have demonstrated a very important role of the natural killer cells in that particular field. So I have a 5 or 10 minutes video here, and I'm going to share it with you, describing the natural killer cells, and it's a nicely animated video. I hope you will enjoy.
Of all the immune system soldiers, natural killer cells are the most aggressive. They are lymphocytes with no immunological memory and are part of your innate immune system. Natural killer cells make up about 5 to 15% of the total circulating lymphocyte population. Natural killer cells are called natural killers because they do not need to recognize a specific antigen before releasing their toxins and destroying a viral infected cell or cancer cell. They target tumor cells and protect against a wide variety of infectious microbes. In several immunodeficiency diseases, including AIDS, natural killer cell function is abnormal, thus allowing the viruses to multiply inside of human cells. Natural killer cells kill on contact. The killer cell binds to its target, aims its weapons, and then delivers a lethal burst of toxins that produces holes in the target cell's membrane and creates events that destroy the cancer cell membrane and cleaves DNA into many pieces in a programmed cell death, that is, apoptosis manner. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. Indeed, research has shown that most of the tumor cells that enter the circulation are destroyed during the first 24 hours, and only a few cells succeed in establishing themselves in distant organs from the primary tumor, which is called metastasis. Natural killer cells may also contribute to immunoregulation by secreting high levels of influential lymphokines. Therefore, research to develop natural products to stimulate the natural killer cell activity, especially in immune-suppressed patients undergoing chemotherapy for cancer, or in any viral disease such as AIDS or bird flu, where immune suppression occurs, is very important in the regulation of aspects of the immune system which affects our overall health. Finally, we come back to the beginning of our knowledge of the natural killer cell. Here lies a natural killer cell in a lawn of sheep red blood cells. These cells are perfect targets for the natural killer cell, possessing no MHC1 receptor. The wrath of natural killer cell is fully vented on the surrounding sheep red blood cells. The The cytotoxins completely destroy the sheep red blood cells, leaving clear plaques around the natural killer cell. There is no apoptosis event, just direct cytotoxicity. The natural killer cell is one of the key soldiers in the fight against viral and cancer growth.
All right. I think I have another video here that also shows, but not as much animation and color. The action of a natural killer cell. So we have here time-lapsed video for a natural killer cell with what looks like a cancer cell here. And as you see, the natural killer cell is injecting the enzymes inside the cancer cell, and now the cancer cell is committing apoptosis, and it will start to fragment and die. So this is more like a realistic video versus the the computer-generated video we watched earlier. And again, to emphasize the importance of the natural killer cells in your innate immune response. Okay, so we move from there into discussing the second part of our innate response. So first we talked about the natural killer cells, and now let's talk about inflammation. Inflammation is a non-specific response to tissue injury or to invasion by bacteria or a foreign target. The ultimate goal is to bring phagocytes, macrophages, and neutrophils, also plasma proteins. Why plasma proteins? Well, plasma proteins have the ability to neutralize, if your plasma already has antibodies, therefore you're bringing in antibodies, you're going to bring in complement proteins, and we're going to learn about the complement proteins in a short bit. And the idea is to isolate and destroy or at least inactivate the invader. When you open the floodgates, you're also washing away the area of infection or damage. So you're removing the debris, and you're bringing that debris, and you're bringing those effectors to the lymph nodes, where T lymphocytes and B lymphocytes are hunkering down, mainly the T lymphocytes, and waiting for some action to be taking place. So inflammation is indeed very, very important. And as I said at the lecture, if you remember the time when you have an infection or an inflammation and or a pimple that is growing, what you have is redness, you have swelling, you have hotness, so it's warm, it's red, it's swollen, and it's painful. These are the criteria that excuse me, these of course are the negative criteria that we remember, but we never realized how important this process in fighting the problem. And if you don't have this inflammatory response, then your ability to overcome that infection will be severely limited. So inflammation is a good thing, unless it goes, of course, out of hand, then we are dealing with a problem. So again, why do we have this kind of exudate or flushing of the tissue? You're going to be moving the foreign material into the lymphatic blood, the lymphatic vessels. You will deliver proteins, whether they are antibodies, or whether they are complement proteins, or whether they are natural antibiotics that you have, or clotting proteins like fibrin. And what the fibrin will do, among other proteins, will create a network, a scaffold around the area of damage, and therefore limiting the infection and keeping it in just one area instead of the infection spreading everywhere. And I compared that with you at the lecture with if you have an attack of some sort, and what you do, or a bank robbery or something, what you do is you seal the neighborhood, and you seal even the city if you have to. And that's pretty much here what we have what the blood or the blood proteins are capable of doing. They seal the area, the infection area, and therefore they limit the spread of the infection, and they isolate that area. Okay, so what triggers the inflammation is that the microbe-stimulated phagocytes, the phagocytes, when they recognize, if you remember the phagocytes, they have the TLR, the toll-like receptors, that will recognize a pathogen-associated molecular pattern, for example, and they will start chewing it. Is that all? No. Once it starts chewing it, then, you know, your phagocytes are going to secrete something to sound the alarm for everyone, so everyone will be aware that there is a struggle going on. The phagocytes will release interleukins, and these are chemicals, mainly paracrine, but of course they have also some distant function to stimulate your bone marrow, such as the colony stimulating factor, as we will talk about later on, or it can induce hyperthermia by modulating your hypothalamic function through the actions of prostaglandin and cyclooxygenase, which produces to you prostaglandin. So the microbe-stimulated phagocytes will release the interleukins, will release colony stimulating factor, and the colony stimulating factor doesn't really have a role here in the inflammation, but what it does, the colony stimulating factor, is that it will swim in the blood and it will go to the bone marrow, and they will order the bone marrow to make more phagocytes, to make more neutrophils by this CSF, which is secreted now by your phagocytic cells. Now, lactoferrin will be also secreted from the neutrophils, and from its name, it quenches iron. And when you quench iron, you make iron unavailable for the bacteria to use it for its metabolism, and therefore you are making the environment harsher for the bacteria. The phagocytes will release the activated phagocytes that got activated because they were interacting with microbes. They will secrete nitric oxide, and the nitric oxide, A, it's toxic, but also B, it induces vasodilation in the arterioles, and therefore the capillaries will open up, and as we said earlier, you have hyperemia in the region, and then that when your capillaries are open or wide up because of this increased flow, then you will increase the exudate or the exit of the plasma protein and the plasma fluids into the extracellular space. As we, if you remember from the studying the factors behind edema, we did understand that the vasodilation will cause the capillary pores to become bigger because of the increase of the blood flow in them, and therefore the hydrostatic pressure, of course, will win the battle, and you have more exudate going into your tissue. Now, your microbe-stimulated phagocytes will also release to you certain interleukins, IL-1, IL-4, I believe, and TNF, I think IL-6. IL-1, IL-6, and TNF, and these will be pyrogens. Pyrogens, from the name, pyro means fire, gens, generators, so they will generate heat in your body. How do they do that? They go to the hypothalamus, and they will activate an enzyme there called cyclooxygenase, and the cyclooxygenase will convert will synthesize prostaglandin from arachidonic acid pathway, and now we have the prostaglandins. The prostaglandins are the one that will reset the clock of your hypothalamus, and therefore the hypothalamus, which is your temperature regulating machinery, will misunderstand the fact and will start to generate more heat in your body, thinking that you are instructing it to maintain your body at a much higher temperature instead of 37 at a 40 degrees or above, and therefore you have fever. Now, as we will see later on, fever is important because A, it will make the life for the bacteria hostile. When you have elevated temperature, the bacterial enzymes don't like the elevation in the temperature too much. And also the process of phagocytosis seems to be better with elevated temperature. So that's why we get the fever. And like I said in the lecture, little fever between 38 and 39, it's healthy. There is nothing to be alarmed about. It's your system trying to eradicate the problem. And but it's only a problem when it goes above 40, 41. Then we should start to be concerned, especially for younger patients. IL-1, interleukin-1, all of these are cytokines, IL-1, IL-6, TNF even. All of them are cytokines. And we have some of these cytokines being secreted by white blood cells to stimulate other white blood cells. So here we have an example of IL-1, interleukin-1 in between the white blood cells. That's what interleukin means. Would enhance the proliferation and the differentiation of both B and T lymphocytes. So we have something that is made by the phagocytes, which are part of the innate immune system, to stimulate the proliferation and the differentiation of the B and T lymphocytes, which are part of the adaptive system. So here we have a nice link between the two. So, as I said earlier, IL-1, IL-6, and TNF, these are the three amigos in a way. These are the same ones that will cause elevation of your temperature, but they will also modify the iron metabolism, just like the lactoferrin did locally. Your liver now, because of IL-1, IL-6, and TNF, tumor necrosis factor, they will modify the iron metabolism in the liver, and therefore there will be less free iron available. And that's very good, of course, because the bacteria is not going to have that luxury of enjoying free iron and growing bigger and healthier as a bacteria. Now, these same cytokines, IL-1, IL-6, and TNF, the three amigos, will stimulate acute phase proteins from the liver. One of them is a very famous one. It's called C-reactive protein. That's a non-specific opsonin, and all of you are scratching your heads now. What's an opsonin? What's an opsonization? As we said in the lecture, if you remember, opsonization, it's a process to make your phagocytosis process more appealing. So to make it more delicious, so your phagocytic cells will like it more to eat that target. The most common way to tag bacteria or an organism for destruction would be by tagging it with a protein, a non-specific protein like C-reactive protein or like the complement system, but also by antibodies. Antibodies are the most important taggers for what we call opsonization. So anyhow, let's go back to this. IL-1, IL-6, and TNF will modify the iron metabolism. They will also stimulate the acute phase proteins from the liver, like the C-reactive protein, which, by the way, we check for when we have someone we suspect that they have, he has acute infection. So we look for the C-reactive protein. They will mediate clotting and later on anti-clotting, clotting for limiting the spread of the disease, but then we have to, after we are all done, we have to remove that fibrin tissue in order to allow regeneration of the tissue. So it mediates the clotting and the anti-clotting. Neutrophils can secrete what we call kallikrein, and the kallikrein will activate a kinine, which is a protein made by the liver, and now you have bradykinin. A bradykinin can stimulate the nerve endings, the pain nerve endings, and it can also cause vasodilation. That is a pathway we can block by Tylenol, for example. And activated kinins can also activate cyclooxygenase, and therefore you can have also prostaglandin. Prostaglandin is another vasodilator, and it also stimulates the pain receptors, so we can block that other pathway using ibuprofen and aspirin, for example. Okay, finally, tumor necrosis factor, which is secreted by the microbe-stimulated phagocytes. It stimulates histamine release and from the mast cells, and that's a very important because histamine is a vasodilator, but of course sometimes things go out of hand and we have too much histamine, and too much histamine will result in swelling. We don't like that much, especially if it's in response to a bee sting or something, and that's where we use antihistaminics to calm down the inflammatory reaction and reduce the swelling and reduce the vasodilation process, as we will see later on, that histamine can also participate in hypersensitivity and an unusual immune reaction, as we will see later on. Okay, so all of these components are part of the inflammation, and this is just a summary here for the opsonization. As I said, you can opsonize a target by an antibody or by complement protein. Here we have an example of C3b, and these proteins will have receptors on the phagocytes. Also the antibody, the constant region here of the antibody, will have receptor on the phagocytes, and therefore the phagocytes now will think this is much more appealing target than just relying on the toll-like receptor interaction with the pathogen associated molecular pattern. What are the pathogen associated molecular pattern? Well, different kind of proteins, different kinds of carbohydrates, so different pattern one way or the other, but now we can enhance that ability of the phagocytes to recognize and destroy the target by tagging that target with either a complement protein like C3b and the C-reactive protein, as we saw earlier, or by antibodies, and we're going to talk about the antibodies later on. In either case, the phagocytes now find it much more appealing to destroy the target and phagocytose and destroy the target. That's what we need to remember, and that's a process called opsonization. Okay, so we do have inflammatory mediators, and we talked about histamine, we talked about nitric oxide, which is not in this particular slide. That is also secreted, the nitric oxide is secreted by the phagocytes. Histamine is secreted on the mast cells, but it was the TNF, the tumor necrosis factor, that stimulated the release of the histamine, if you remember. We have blood proteins that are coming now because the capillaries opened up. We have the kinins, and the kinins will be activated to become bradykinin. We have the prostaglandins that are synthesized by an enzyme called cyclooxygenase, and we have other proteins. All of them are part of what we call inflammatory response. Okay, so we are at a war zone. We have inflammation, we have bacteria that came in. How do we bring more phagocytes? We need to sound the alarm to bring the SWAT teams to, and also we need to open up the capillaries. How does that happen? So let's look at the slide that demonstrates it in a picture. They say pictures are better than thousands of words. So here we have a splinter that brought in some bacteria, and the bacteria will go at the site of injury and they will become phagocytosed by the macrophages here. The macrophages will start to release cytokines, and those cytokines are going to interact with the endothelial lining of the blood vessels or the capillaries. And when you do that, two things will happen. The pores in the capillaries will get bigger, but also the capillaries will start to produce a protein called cell adhesion molecule. Now the cell adhesion molecule is very, very critical because it will sort of act as a glue trap, literally as a glue trap for the neutrophils that are walking by minding their business. They're supposed to be like tumbling around, and this is supposed to be extremely smooth. But you can imagine once you start having the very sticky surface, and that's because of the cell adhesion molecules which got stimulated by the cytokines, these will not tumble anymore, and instead they will get stuck to the endothelial lining, and then they will start to migrate in a process we call diapedesis. Then you have tons of neutrophils in the tissue, and that's a very important way to get more fighting power to the site of inflammation. Is it only limited for neutrophils? No. At the same way, you are also stimulating the immigration of your monocytes, and as we said earlier, the monocytes will become macrophages, and this is a very important process to replenish the macrophages and also to bring about to bring in more neutrophils, and the neutrophils are indeed your slayers. They will be chasing the bacteria, as we saw earlier in the video, and destroy those bacteria. Other things happen. The cytokines will also talk, like TNF will talk to the mast cells. The mast cells will produce histamine, and the histamine will also cause vasodilation and of your arterioles. Remember, vasodilation doesn't happen in the capillaries, but it happens in the arterioles upstream, and that will result in more blood flowing in the capillaries, and that of course will flow more neutrophils in, but will also bring in proteins, and we discussed those proteins, the blood proteins, how important they are in fighting the inflammation, or the source of inflammation. Okay, so far we discussed two different components of the innate response. One of them was our natural killer cells, and the second is inflammation. At the end of the inflammation, sometimes there is a tissue repair. In most cases, of course, we have tissue repair. Sometimes it's imperfect if the inflammation was extensive, and you will end up with some scar formation, especially in areas that do not have the power to regenerate. Of course, we have drugs that can suppress the inflammatory process. Non-steroidal anti-inflammatory drugs like aspirin, ibuprofen, celecoxib, all of these guys are capable of inhibiting the inflammatory process. Glucocorticoids are much more aggressive. They will essentially inhibit all your immune response and not just the inflammatory responses. And because they do so, we have to be very cautious and very careful prescribing the glucocorticoids, of course. We will study the glucocorticoids when we get to the endocrine system. Mainly the cortisol is what we're talking about here. And they will reduce the ability, as I said, to fight infection. They induce lipocortin. They will inhibit COX-2. If you remember, COX-2 was the reason we have prostaglandin, and we do understand and appreciate what prostaglandin does for you. They will also inhibit the T-cell proliferation, and therefore you have much lesser immune response. So it's very important to realize that glucocorticoids are not trivial, and therefore you need to be very, very careful if you become in a situation where you need to prescribe something. You need to be cautious on the overprescription of glucocorticoids. Okay, so we covered two components, two major components. Now we go into interferons. So the interferons really are They are secreted by normal cells. Don't misunderstand and think that interferons are only made by viral infected cells. No, interferons are made by normal cells as well, and they have other functions to warn your system and to activate, for example, the natural killer cells, as we will see in a short bit. But in a viral infected cells, they become extremely important. Why is that? Because when a cell is infected with viruses, then the cells as a byproduct will make more of these proteins, of the interferons, and those interferons will go to uninfected cells, healthy cells, and will bind to specific receptors and will warn these cells about the fact that there is a virus on the loose. It's like an alarm system that will say, be careful, we are being under attack. So these healthy cells now will make virus blocking enzymes. Essentially they are nuclease proteins that are capable of destroying the nucleic acid of the virus if it gets injected into the cytoplasm. And they're waiting around, but they're inactive. They will become active, however, if the cell gets subjected to virus infection. And now these enzymes that are hunkering down here will interact, will be activated, and they will interact with the nucleic acid, therefore preventing the nucleic acid from being integrated into the cells. So it really protects the non-infected cells or the healthy cells against future invasion by the virus. And they are mainly produced by infected cells, although interferon is also made by other cells. So, for example, lymphocytes are also made by, also are capable of making gamma interferons. Many of the white blood cells, they make alpha interferon. Fibroblasts, they make beta interferon. You don't have to remember those types. You just need to understand that interferon is made by several types of white blood cells, and it's extremely critical. It's not just made by a virus infected cell. Now, the interferons are known to activate macrophages, make the macrophages extremely aggressive. They're also capable of activating and mobilizing natural killer cells, and therefore we started now to utilize the natural killer cells in fighting against cancer. Remember, the natural killer cells are the ones that seek and destroy cancer cells. So when we give genetically engineered interferons to patients, we are indeed stimulating natural killer cells, and therefore we have much more power now to fight against cancer. And therefore interferon now, as you will see in the next slide, is being now sought as a treatment for hepatitis, genital warts, even in cancer. Why? Because it is capable of stimulating and mobilizing the natural killer cells. So there are many trials with interferon that blocks and treats hepatitis B virus, and the success stories are amazing. And so interferons are, of course, major players in your innate response system of your innate immune system. So we covered three things so far. If you try to remember with me, we talked about the natural killer cells, we did talk about inflammation, and we're talking now about interferons. That leaves us with the complement system, the complement proteins, and this will be our next slide. So essentially the complement system and the complement proteins, these are made in the liver, they're inactive, but they can get activated, and the way they become activated either by the presence of an antibody on the target, okay, we'll call that the classical activation, or by a different pattern like carbohydrates or something on the target that will activate your complement system. But you can also appreciate the fact immediately that it will become much faster activation if there is an antibody already coating that target. Okay, fine, we established that. So what do the complement system proteins do? Well, once they become activated, they can do two things. The major thing they can do is to tag the bacteria, or tag the target for destruction. Remember what we call that? We call that opsonization. And the other thing they're capable of is to form what we call membrane attack complex. If you remember, they make that tube and they insert it, it gets inserted into the cell membrane or of the bacteria, or the cell membrane of the cell that they need to attack and destroy. And therefore now you're connecting the cytoplasm inside with the outside surrounding, which is the extracellular space, and essentially you're killing the cell at that point. The cascade, you don't have to remember C1 activates C2, C activates C3, you don't have to remember that, but you need to understand and appreciate that the complement system is part of your innate response. It's non-specific, and it's activated either by the presence of antibody, we call that the classic activation or classic complement fixation, or the non-classic, which involves just recognizing different pattern on the bacteria or the cell that you're trying to destroy. I have a picture here of the CAM, the MAC, I'm sorry, the membrane attack complex, and we have proteins here of the complement system, and when they get inserted, as you can see here on this bacteria, you can immediately appreciate the fact that this bacteria now is going to lose all its cytoplasm to the extracellular space, and therefore it's going to die. And that's the beauty, of course, of the complement system. I don't want you to think that the MAC, the membrane attack complex, is the main mechanism through which the complement system is helping you, aiding you in your innate immune response. It's not. I mean, it's cool and stuff, making a tube, and the tube is connecting the inside with the outside, but that's not necessarily the main function for the complement system. The main function is the opsonization, and therefore you are tagging the organisms, tagging the targets for destruction, and they will become more appealing for phagocytic cells to destroy those. Okay? All right. Other functions of your complement system, these are the main two functions, opsonins and the MAC, but they do other things. They act as chemotaxins. They bring in, they are inviting white blood cells to come into action. That's what we call chemotaxis. And they will promote vasodilation. They will stimulate the release of histamine, which is a good and bad thing. I mean, if you have excessive stimulation of histamine, you can end up with an anaphylactic shock, for example. So we have to watch out for excessive activation of histamine, of course, from the mast cells or the basophils. The activated complement system is capable also, or the complement proteins that become activated, they are capable of activating kinines. And if you remember what happens when you activate kinine, you end up, for example, with bradykinin, and the bradykinin is a vasodilator, but it's also a very potent pain stimulant for the nerve ending. So all of these are functions by the complement protein and the complement cascade, the most important of which, of course, is acting as an opsonin and making the MAC, the membrane attack complex, but they're capable of other things: chemotaxis, vasodilation, stimulation of histamine release, and activating the kinines. Okay? So I'm going to stop with this video at this point here, which will conclude our innate immune system. We did discuss the components of the innate immune system. We will revisit the macrophages in the second or the third video, where we will talk in greater details about the macrophages and about how they share in what we call antigen presentation, and how important that is in the process of activating the lymphocytes, as we will see in our next video. So we did describe the innate immune system. We did talk about the players. So now in our next video we will talk about a very interesting mechanism for our system to fight disease, and the role of the lymphocytes, and how the lymphocytes from the adaptive system can crosstalk with players, whether these players are the natural killer cells, which are also lymphocytes by the way, or with neutrophils, or with macrophages, and they talk back and forth. This one activates the other, and in reverse as well. And we will see how the two systems, the innate and the adaptive immune responses, can become integrated, and they collaborate together in order to eliminate the problem for you. Okay, so watch the next video, and hopefully you'll find this one helpful in your review process. Have a good day.